Hello, everyone, and welcome to our Pediatric Nephrology Fellowship webinar. Today, we have very special event because this webinar is in collaboration between the World Kidney Academy and the European Society of Pediatric Nephrology. So again, thank you so much, Dr. Neville Soliman, for organizing this webinar. And today we have very special speakers and moderators. So we have Dr. Rick Westland from Amsterdam, Alina from Germany, and Julia also from Germany. And these three are between speakers and moderators, panelists, and they are representing the ESPN, the European Society of Pediatric Nephrology. Myself and Dr. Niveen and Dr. Khalid are representing the World Kidney Academy. So very nice collaboration. Here is a brief agenda of our meeting today. Again, Alina is going to introduce Julia, who is going to give about a 20-minute presentation on monogenic causes of CAKUT. Then Dr. Niveen is going to moderate the discussion. And the next speaker will be Dr. Rick Westland, and he's going to focus on the complex mechanisms and understanding of genetic background of CAKUT. Then Professor Khalid with Elena is going to handle the question and the answer with Rick, and Professor Niveen is going to give the closing remarks by the end of the webinar. Again, we are almost finishing module 1 of the Pediatric Nephrology Fellowship. Our fellows are going to start their exams next week, so good luck. And we are very happy that the World Kidney Academy and the IBNA, the International Pediatric Nephrology Association, and also the European Society of Pediatric Nephrology have agreement with MOU for collaboration. Again, in this fellowship and our other educational activities, we are focusing more not on a monologue or one-way direction education by giving lecture or giving readings, but we are trying to involve and engage our fellows with the audio visuals, demonstration, discussion, and let them also to present cases. This fellowship is structured as 3 modules, 16 weeks each. It's a real education, not based only on webinar and seminar, but there will be a lot of journal club, MCQs, clinical case scenarios. So it's bi-directional, and it's not a monologue, but it's a dialogue. Upon completion of the fellowship, the fellows should expect to be an expert in the PEDNEF field, and also to graduate from a trainee or mentee to a trainer or mentor level, and also receive 96 credit hour accredited graduation certificate from the American Association of Continuing Medical Education. Again, all of these activities, we are trying to improve our pediatric nephrology patients' outcome and the standard of care, because knowledge itself has no value till we put it in practice. And the great aim of education is not the knowledge itself, but the action. And with that, I'm going to stop by here, and I will let Elena to take over and introduce Julia.
Wonderful, thank you. So as part of the ESPN CAKUT UTI and bladder dysfunction working group, it is my great pleasure to introduce our today's speakers. And we will start with Professor Julia Höfele. She is head of the ESPN CAKUT working group. And while she worked in several human genetics institutes, including the laboratory of Friedhelm Hildebrand, she is now professor of human genetics at Ludwig Maximilian University in Munich in Germany. And she is there focusing on renal genetics. And with her multiple studies of CAKUT patients, she has contributed significantly towards the current knowledge of CAKUT genetics. And together with Professor Weber, a pediatric nephrologist from Marburg. She is hosting the EURECA registry, an international registry combining genetics and clinical data to gain the knowledge in familial CAKUT cases. And hence she will tell us or give us a talk today about monogenetic causes of CAKUT. So, Julia, it's a great pleasure.
Thank you very much, Alina, for these nice words, and it's a great honor for me to be part of this webinar. Just a second. So you can see my presentation, I think. Rick Westland and I made the title The Genetic Architecture of CAKUT for this complete webinar. So first of all, I would like to show you the spectrum of genetic inheritance. If you look on the left side of this figure, you can see that we have probably a gene with a variant inside, and this specific gene with this variant leads to a specific phenotype. This is very rare, and you only have a selected genes, and if you have this variant, it leads to this phenotype. So this is called the monogenic or the Mendelian inheritance. If you look on the other side of this figure, you see here, I think you see my arrow, you see multiple genes, and if you have here several variants in there, you have a so-called oligogenic or polygenic inheritance. But if you have, in addition, some other factors like environmental factors or smoking, radiation, or viruses, or so on, and they could lead to additional variants in these genes, you have a multifactorial inheritance. So you have genetic and non-genetic factors which lead to a phenotype. But this phenotype can be pretty different because you have different factors which can influence this phenotype. So this phenotype is not specific. It has a broad spectrum. So these variants are pretty common in the genotypes. So it's a very complex inheritance. But in my part of this presentation, I will focus on the monogenic or the Mendelian inheritance in CAKUT specifically. So I will give you a short overview about the monogenic background, and afterwards Rick Westland will show you the complex genetic etiology of CAKUT. So let's start with my presentation. So CAKUT, as you know, has a very broad phenotypic spectrum. And we have also a variable expressivity. As you can see here on the right side of this figure, there are lots of different phenotypes focusing on the kidney, on the bladder, and on the ureter. So we have some milder forms like a vesicoureteral reflux, a grade 1 or 2, but we can also have a severe form like bilateral renal agenesis, which is not compatible with life. Sometimes we have different CAKUT phenotypes in one individual, and then we call it complex CAKUT. You should know about this term. We know already that up to 50% of all pediatric patients with CAKUT develop kidney failure. So they will need dialysis and kidney transplantation. Additionally, in some of these individuals, we also see extrarenal manifestations, and we then call it syndromic CAKUT. I will show you some extrarenal manifestations later in this presentation. The frequency is pretty often of CAKUT. We see it in 3 to 6 per 1,000 live births. So it's not a rare disease. I show you here a figure which is from a publication from 2009 from Manolio. And you can see here on this figure, if you have rare variants in the general population, then they have a high effect size. So if you have this variant, you definitely have a phenotype. If you have other variants which are very common in the population, then the effect size of them is pretty low. So sometimes you see the phenotype, but the phenotype can also be quite different in one individual and the other. So it depends. If you have rare variants, then the phenotype is specific. If you have common variants, probably some of the individuals do not show any phenotype. I will focus on the Mendelian or the monogenic forms, and this is always with rare variants or rare alleles combined. What kind of genetic causes we have and inheritance patterns if we focus on the Mendelian inheritance? This is a picture from Rick, or one of Rick's publications. And we have different inheritance patterns. The most common one for CAKUT patients is the autosomal dominant form of inheritance. So you have one affected allele or one affected gene where there is a disease-causing variant, and this leads to the phenotype. The other form is the autosomal recessive one, where you need to have two variants, so a biallelic form. And so either you have compound heterozygous or homozygous variants in one gene from inherited from the father and the mother, and then you develop the CAKUT phenotype. Sometimes you see an X-linked form, so mainly males are affected because they have a hemizygous variant in one of a gene which is located on the X chromosome. If you want to look, so far we perform exome sequencing or genome sequencing, or in some specific cases where we probably know the familial variant, we perform Sanger sequencing just for targeted sequencing. I will explain you the differences also later in this presentation. So far, we know more than 50 genes which lead to CAKUT, or disease-causing variants in these genes lead to CAKUT, but only probably 12 to 20% of the affected individuals show disease-causing variants in the so far described genes. So only a small portion of the patients show a monogenic cause so far. The inheritance patterns I already explained to you. In a handful of patients, we also see mitochondrial defects, which I did not mention so far. Autosomal dominant forms usually develop, can be inherited by one of the parents, so either the father or the mother, or they can also be de novo. So it means it's only in the child, but not in the blood, the variant of one of the parents, so either in the father or the mother. We have different types of variants, the so-called single nucleotide variants or copy number variants. This is the acronym. So let's explain these two types of variants. First of all, the single nucleotide variants. Here in the first line, we have the wild type, so the starting sequencing. You see here this A, and if this is not here, the A, it's a deletion. So you get a frame shift, and normally after some of the nucleotides, you also have a stop codon. So on the other form, the A can be changed into a C. This is called a substitution. So the amino acid, which will be encoded by 3 of the nucleotides, will change, but the rest of the protein will be normal. But this is usually leading to a milder form of the phenotype. Then there's also one which is called insertion. You see here that the A is one on the right side and the T is in the middle. and this T should be not there, so you have the T inserted, so you also get a frame shift, and usually you get a stop codon, which also means that the protein is not in the full length, so it's shorter. The other form are the copy number variants. You see here in the middle of this figure, the normals or the wild types. You see 2 chromosomes from the same, and we see these regions A, B, C, D, and we focus on the D. If you go on the left side of this figure, you see that on one of the chromosomes, part D is missing, so we call it a deletion. And if you go on the right side of this figure, you see that the D is duplicated on one of the chromosomes, so we call it a duplication. These both also can develop a phenotype or can lead to a phenotype in our presentation here for a CAKUT. So common CNVs, so copy number variations, are described or showed here in this table. A handful of copy number variants. I think the most common ones is the deletion and sometimes the duplication on the long arm of chromosome 17, and the HNF1B gene is there included. I think everybody of you know about this gene. The other one is on the long arm of chromosome 22. Here also you have, in most cases, a deletion, and the symptom is called the DeGeorge syndrome, which is associated with mental retardation, heart defects, immune deficiency, and also renal malformations. How about extrarenal manifestations? I already told you that we have about 50 genes which are known so far involved into CAKUT, and if you have variants in some of them, you can also develop extrarenal manifestations. As you can see here in this picture from Kagan et al., there are extrarenal manifestations all over your body. For example, central nervous system, where you can develop mental retardation, or if you have other genes involved, you can develop hearing loss or hearing impairment, or dysplastic ears. Or on the other side, if you look on the eyes, you can develop a retinitis or colobomas, or you can develop heart defects, or endocrine malformations, or which is also very known, are defects or malformations of the limbs, so on the hands or the feet. So the most frequent genes which we see in the literature and in our patients is the HNF1B, which has disease causing variants, or PAX2 or AR1. I would like to present you 2 cases or 2 families which I saw in our department. The first one was a 6-year-old girl which had proteinuria, and she had an increased creatinine, and she also had a hearing impairment. In the ultrasound, we could see, or the pediatric nephrologist could see, bilateral kidney hypoplasia. And she also described, or her parents described, that she had bilateral ear tags, and she had also ear dysplasia. Her father had a hearing impairment since childhood, and he told us that he had an operation as a baby, and that it was at the anus because it was very small. And he also had a chronic kidney disease since the age of 35 years. Here you can see some pictures of their hands. These are the hands of the father. On the right side, you see the hands of the girl. And on the lower part, you see the ear of the father and the ear of the daughter. So if you look on the hands from the father, you see that the thumb is pretty long. And you can see it even better in the girl, that it's very long and it looks like a triphalangeal thumb. The father told us that he knew about his thumbs his life long, and he also told this the nephrologist, but nobody cared about that. Then we looked on the ears, and you see here it's a little dysplastic, but it's very small. But if you look on the girl, you see it's very dysplastic, the ear, and you can see here the scars where the ear take was. So we performed molecular genetic diagnostics, and we could identify in the girl a heterozygous pathogenic nonsense variant in SAL1. SAL1 is associated with the so-called Townes-Brocks syndrome, and this is associated with stenosis of the anal canal, which would fit very well to the father's medical history, also with the ear dysplasia, thumb malformations, the renal hypoplasia, which we also had in our family or in our affected individuals. It can affect the heart, and some of the patients also show mild mental retardation. The inheritance pattern is autosomal dominant, so you have a recurrence risk of 50%. The father was also carrier of the variant. I mean, because of the story, it fits well. Okay. What we see in this family, that the phenotypic variability was pretty large. I mean, the father had the CKD at the age of 35. We think that the girl will develop this quite earlier. And we also had the ear malformations in both, but it was quite different. And when we expressivity, we can divide it into two types: the intra-individual variability, where we have the co-occurrence of different anomalies within the same individual, or we have the inter-individual variability, where we have different CAKUT phenotypes in individuals with the same disease-causing variant. So, for example, if we have one family, they all have the same variant, but they have different phenotypes. And also what we see is an incomplete penetrance in CAKUT, depends on the gene. I would like to show you a second family we already published 1 year ago. Or a 12-year-old male individual with syndromic CAKUT presented at our department. He had bilateral hypoplastic kidneys and a chronic kidney disease, and he's already kidney transplanted. And he also showed craniofacial dysmorphism. He had problems to open his mouth, to eat, to speak. And he also had a small mental retardation. His parents were consanguineous, and I show you here the pedigree of the family. This is the affected individual. His parents were consanguineous. The parents of the mother were also consanguineous, and here's also a family, or the sister of this affected. So the aunt was also consanguineous with her husband, and they also had 3 affected children. They had the same phenotype. One was already died, and there were 2 affected left, and these both are also kidney transplanted so far. And we saw this girl, and she also had the problem to open her mouth and to eat and to speak. So we performed exome sequencing. It was several years ago, and we could identify no disease-causing variant in known CAKUT genes, but we found one homozygous frameshift variant in, at that time, a candidate gene in the FOXD2. And we searched for other families and could also find one family in Turkey and one family in Israel, which also had homozygous variants in FOXD2 and which also had a CAKUT phenotype. FOXD2 is a 1 exon gene, so we only have 1 coding exon. and it's encodes for a transcription factor, which is very common. And it's known that there's a knockout mouse model from 2000, and there the mice had kidney hypoplasia, which fitted very well, but they had a reduced penetrance. We also know that this transcription factor is involved in the development of the tongue and the mandibula, and this would fit very well to the phenotype of our patients. So the mouse model from the 2000 was not available anymore, and so we generated a new mouse model at the German mouse clinic at the Helmholtz Zentrum in Munich. And as you can see here on the right side of this slide, here on the left side, you see the wild type mouse, and on the right side, you see the mutant mouse, and you see here that the papilla is atrophic in the kidney. And we also looked on the head of the mice, and as you can see here, the gray one is the wild type mice, and the red one is the mutated mice or mouse. And you see the mandibula, and when you compare that, you see that the red one is smaller and that it's completely different from the ankles and the length of these ankles. So this also fits very well, that these mice also had problems, not to speak, but to eat, which fits to our patients. Okay, this was the story, and I think it's very important that to perform sequencing in your patients, especially if you have familial cases or syndromic cases, because it's or the detection rate is pretty high, that you find a disease-causing variant or probably a candidate gene. But what are the differences in these sequencing techniques? I told you already that we have different possibilities to sequence. And if you look on this apple, you can compare this like the genome. You look on all variants which are in this complete thing. So if you have your DNA, you look on the complete DNA and the coding and the non-coding variants. And this is called the genome sequencing. The exome only looks on the coding variants, and it's about 1 or 2% of the whole genome. So you only see parts of that. But as we know at the moment, a high portion of the disease-causing variants lie within the coding region. So probably this examination would be a first step to examine in your patients or to perform in your patients. If you only look on genes, which are already described in the literature, then it's called a gene panel, multi-gene panel diagnostic, whatever. The disadvantage is that if probably tomorrow there's a new gene described in the literature, this will be not included in your examination. But if you have an exome or a genome sequencing, you can always go back to your electronic data and you can look, are there any variants in this new published gene? Here, the different sequencing technologies are shown again. If you have a known familial variant, you usually perform a Sanger sequencing because you need only to look on one point of your genome. So it's a pretty simple, cheap, and fast technique. If you think it's a CNV, you use a karyotyping, a microarray, or a SNP array. But otherwise, I would recommend to use a short-read next-generation sequencing analysis, probably the exome or the genome sequencing. And I think what will be next in diagnostics, at least in Europe, will be the long-read sequencing. The advantage is that you also can look on repeat expansions, which is probably not that common in nephrological diseases or kidney diseases, but for example, in neurological disorders. The only one I know in nephrology might be the ADTKD, where you have an expansion or an insertion in the MYC1 gene, which you could cover with long-read genome sequencing. But the disadvantage is when you have the genome sequencing, you get lots of variants which you have to filter and which you have to analyze. Are they really disease causing, or it's just probably a benign or likely benign variant? Usually if we, for example, have an exome sequencing, we have more than 20,000 variants in the beginning, and then we have to filter down to look what is the real variant which is disease causing. So we look for, first of all, what is in the population, and then we filter it to all variants which are less than 1% in the population, and then we look again in this rest of the variants, what is the function? Is this a loss of function or a splice site variant? And then we look also, are they already described or are they phenotype related? And so probably at the end we have 1 to 3 or 5 variants left, and then we have to look on them by hand. There are several filtering softwares or softwares where you can examine your exome or your genome, for example, Imagene from Illumina or Varvis or Varfish. In 2015, the American College of Medical Genetics and Genomics published recommendations which are now used worldwide because you can classify a variant and you can reproduce it everywhere. So this is very easy. If you have a variant, for example, from another country, you can look again and you get more or less the same result of classification. There are several updates from 2019 and 2024, and at the time it's used all over the world. So this classification groups variants into 5 classes. Before 2015, all variants where we thought they are disease causing were classified just as mutation, but this term should be avoided now. So we have these 5 classes. We have the benign, which means that we think it's 99% sure that it's benign and does not lead to a phenotype. And the likely benign, where we think it's 90% sure that it does not fit with the phenotype. Then we have the variants of uncertain significance. We do not know, are they benign or are they pathogenic? But probably this class will change over the years if probably you get or you get involved or you find other patients worldwide which have the same variant, then probably upgrade of this variant is possible. On the other side, you have the likely pathogenic variant. This means that you have a 90% that you are 90% secure that this is the disease-causing variant, and the pathogenic where you have the 99% secure that it's disease-causing. On these both, the likely pathogenic and the pathogenic are called disease-causing variant. So when should you perform genetic testing? We think, and it's also described in the literature, that the individual should be affected on both sides, or if it has exogenous manifestations or syndromic cases, or if you have a positive family history, or if the family is consanguineous, then you have a higher detection rate than if it does not fit in one of these terms. So we published one CAKUT cohort. It was almost 100 individuals, and as you can see on the upper part of this figure, there are the bilateral kidney affection individuals. We could identify in 8 individuals of them disease-causing variants, but the high proportion of the individuals with CAKUT did not show any disease-causing variant. So Alina already mentioned an ongoing ESPN study, so-called the EURECA registry. It's a registry on familial CAKUT cases where we collect clinical data as well as genetic data. But if you only have the clinical data, these patients are also very welcome, and we try to perform exome or genome sequencing in the individuals which did not have any genetic analysis so far. This will be covered by research grants. So the main persons are Stephanie Weber and I, and we also have a steering committee with some additional people. So Dominik Müller from Berlin, Anja Büscher from Essen, and Alina here from Erlangen. Unfortunately, although we published this at the ESPN, nobody want to join us, but if you are very welcome, and we would like to see additional patients also from your countries. So far there are more than 120 individuals in there with the clinical and often genetic data, and they all have familial history. We don't need both or more affected individuals, but they should have the medical history that they are familial. And some of them are isolated, but we also have some syndromic phenotypes in there. And we plan to make this registry as a long-term follow-up over the years. So please include familial CAKUT cases. You either can go directly on the website or you can contact me just by mail, and then I can give you an account, and then you can upload your patients. So as a take-home message, as you already seen in my presentation, there are several monogenic causes, but we have lots of additional factors which lead to CAKUT, like environmental or epigenetic factors. Probably Rick will tell you about these. And so far, monogenic causes are pretty rare, about 20%, but I think it's still worth to perform genetic analysis in your patients. But the higher detection rate will be in the bilateral or in the syndromic cases or familial cases or in individuals which come from a consanguineous family. So the phenotype is highly variable, and we don't know so far what is the reason for, but probably there are other genetic factors or non-genetic factors which influence the phenotype. But I think genetic testing should be offered to your patient because sometimes you can optimize treatment and you can give adequate genetic counseling. And I think also it's very important, at least for me as a human geneticist and a pediatrician, to work together with other pediatricians or pediatric nephrologists and nephrologists to optimize the treatment of the patients. So thank you very much for your attention. This is my group. Corbinian Riethammer is at the moment at Friedhelm Hildebrand's lab, working on research basis. He's a nephrologist and human geneticist, and Matthias is nephrologist and have several doctoral students, and here are my collaborators. Thank you very much.
Thank you very much, Julia. That was elegantly presented. Thank you for this. We had a couple of questions, though. I think one is answered. When do we need to look for genetic testing in CAKUT? And you already presented this. And the other thing, what's the role of variant of uncertain significance? For example, I think one of the participants of the fellow said PAX2 VUS. So what's your take on the
I think that's
VUS?
pretty difficult to answer. I think at least in Germany, we only tell the family if we have a hot WUS or a warm WUS. We have a classification in between this WUS, where we have additional classifications in between. If it's more located to the benign side, then we don't tell the family. If it's on the other side to the likely pathogenic, we tell the family, because we think probably if we find other individuals worldwide, or if we perform further analysis or transcriptomics or proteomics, then we could probably put it in the likely pathogenic. So depends on several factors, I think.
And there's another question. I think this is needs clarification from your side. And there's a mix-up between monogenic and polygenic. You presented that, but they think that monogenic should be one gene, but one gene of a set of genes. Could be more than one genes affecting this phenotype, but it's only the variant or the disease-causing variant is in one gene, versus polygenic, and they are asking about epigenetics.
Exactly. I think Rick will cover the polygenic factor. We will see that later.
Okay.
Exactly, but I told you the monogenic form, one gene, one effect.
Okay.
Or probably, yeah.
Okay, thank you.
Okay, if you want EURECA, just I can give you in the chat the website and also my contact information. Then you can just write me an email.
Thank you. Thank you.
Okay, then probably we will hand over to Rick,
Alina?
I think, or?
Yeah, so the next talk will be given by Dr. Rick Westland, and he is a young pediatric nephrologist from Amsterdam, and he is actively involved in the ESPN CAKUT working group as a board member, and after his postdoc time in New York, where he joined Simone Sanacchieri's lab, he has actively continued CAKUT research, and currently is leading the Art Deco project, and will tell us about the complex genetics. And now we heard from Julia about the monogenic part, and we'll now hear more about multifactorial genetics of CAKUT.
Thank you, Alina, and thank you, organizers, for this wonderful opportunity to talk about one of my favorite topics in pediatric nephrology. And thank you, Alina, for calling me young. That's a big compliment. So thank you so much. So yeah, Julia gave the perfect introduction, I think, into genetics of CAKUT, and you might see some overlap here because the complex genetic etiology is not so easy as the name already intends. So as Julia said, CAKUT has a lot of heterogeneity, right? So there's a heterogeneity in phenotypes. It can be because of the changes in kidney tissue, in number, size, or position. But this can also affect the ureter and the bladder, and even the urethra or the lower urinary tract. So there are very many phenotypes, and some of them are rare. Think about posterior urethral valves or bilateral kidney agenesis, whereas others are quite common, as Julia already said, like a low-grade VUR or transient hydronephrosis, which we find mostly antenatally right now, or duplex kidneys. It actually happens one in about 200 to 500 births, so not that rare at all. And this may also get you to think about the etiology of CAKUT, right? Because if you look at these very rare deleterious monogenic forms, it would not fit a phenotype like low-grade VUR, which is quite common. So this means that although we use this umbrella term CAKUT for this entire spectrum, because we know there are several connected etiologist standpoints or points of origin. It might not always be the case that that they are completely the same. So it keeps us on our feet, and we have to be creative to unravel this etiology. This figure, I like it a lot, and Julia also showed it, is I think a good summary of my talk at slide number 2. So these genetic causes, Julia talked about monogenic disease and also a little bit about structural variation. I will show you some data on this in more detail as well. But there's also these common variants, which are identified by large-scale genome-wide significant studies, association studies, which have not been performed in CAKUT as much. And I will tell you today why I think this is. And of course, we want to know, even in the audience, about epigenetic causes, which has to do with the accessibility of DNA and modifying translation of the DNA into proteins. But epigenetics in CAKUT is quite hard because we rarely have the opportunity to have tissue, right? So to look straight into the kidney tissue itself, we almost never have this, because a kidney biopsy in CAKUT is a relative contraindication. So it has been really hard to find these epigenetic causes, because you cannot derive these changes in the kidney tissue from, for example, leukocytes in the blood. And then there are these environmental causes, which we think has to do a lot with, for example, maternal health, right? So kidney development takes place exclusively during gestation. So it is well feasible that if the mother experienced some environmental hazard, for example, by diseases such as diabetes mellitus or obesity, then the chances of getting a child with CAKUT is higher. So we have seen this in mothers with diabetes, for example, but we do not know the pathomechanisms about it. And the data is also quite contradictory. For example, we know that using folic acid may protect against CAKUT, whereas the multivitamin use, which also contains folic acid, has shown a higher association of getting a child with CAKUT. So it's all about unraveling these pathomechanisms to better define these environmental causes and also their interaction with the genome. Because, you know, the genome is not static, it changes depending on the environment and epigenetics, and it is really hard to pinpoint where these changes are and how they occur. Some other things that may influence kidney development are, for example, nephrotoxic medications, if the mother is taking them, but also if a child is born prematurely and gets medication, for example, for patent ductus arteriosus at the NICU. This may also disturb nephrogenesis and lead, for example, to a sort of acquired form of kidney tissue malformations. As Julia already said, we can only explain a minority of patients based on single nucleotide variants, 10 to 15%, depending a little bit on the cohort characteristics. And then we know that CNVs also take about 5% of these cases additionally as well. But that means that the vast majority of patients, they don't have a clear-cut genetic cause. While we see that familial occurrence is quite high in these patients still, we cannot really pinpoint the cause. So this might be a combination of those. And this means we have to look into more complex pathomechanisms of disease. And Julia already says a little bit something about variable expressivity, which is, I think, one of those mysteries in biology, is how come that if you have the same variant, the phenotype can be completely different, right? This has to do with how the DNA is translated, but also how it is read. And also modified by other factors which we don't know. So these are the things and these are the challenges we have to explain better to the patient how they become diseased, but also it gives opportunities to connect the etiology to the prognosis, so we can actually start improving the prognosis of children with CAKUT. Right, and this is a little bit of a image that summarizes the difference between incomplete penetrance and variable expressivity. So CAKUT also is very well known about incomplete penetrance, which means that you carry the variant, which is a pathogenic class 5 variant, which is not de novo but runs through the families, but there are multiple family members who carry the same variant but don't have the phenotype. How this happens, we don't really know in most of the cases. Whereas variable expressivity means that you carry the same variant in your family or between families, for example, but the CAKUT phenotype is completely different. And these are hallmarks of genetic CAKUT. And this is also the reason why gene discovery studies are very hard to perform, because if you want to prove causality, as Julia showed with the FOXD2 variant, it's really hard if you have incomplete penetrance to prove your point, right? You need to go to the mouse model to see that they actually have a kidney phenotype or, for example, a jaw phenotype, as Julia said. But this also means that there might be a role for poly or oligogenic etiologies, which leads to this variable expressivity or incomplete penetrance. And we're going to zoom in a little bit on this better. And my colleague in Utrecht, Albertine van Eerde, a very well-known nephrogeneticist, already showed that compared to other kidney diseases, the diagnostic yield, so finding the genetic cause of the disease in CAKUT, is very low compared, for example, to very well-known ADPKD or also to tubulopathies, which have a much higher yield for a clear monogenic cause. For CNVs, it's a little bit better compared to other kidney diseases. But then if you also look to the recommendations that for genetic testing in CAKUT, as Judy already said, for example, if there's a positive family history, you see that CAKUT as a kidney disease still has, although the positive family history is quite high, it still has a low diagnostic yield. So it doesn't perform that well. And the same applies for extrarenal features. If there are extrarenal features in CAKUT, the diagnostic yield is not so high as, for example, for ADPKD or ciliopathies or glomerulopathies. So this means that going on a quest to find the genetic cause in CAKUT, you have to be a little bit mindful and careful with what you see. You have to prepare your patient also that the chance that you find something might not be as good as you hope. And also this is a call to arms, I think the same as Julia said, that you have to do this together with your geneticist, preferably, so you can actually quantify or qualify this variant a lot better and also explain the consequences to your patient. So this is an advice to do in the clinic if you are having a child or a person you suspect of genetic CAKUT. So copy number variants, they are also in this complex range of genetics, which you can identify with genome-wide microarrays. This is still the cheaper and faster option, but I think this will all be changed into whole genome sequencing soon, because the costs of genome sequencing are getting quite lower, and you can do the single nucleotide variants and the copy number variants with only one modality, so that's nice. But if you have, for example, a deletion that takes out the gene, you have this monogenic disease, the gene that drives the CAKUT phenotypes, but the gene can also be interrupted or fused or changed position because of the CNV. So CNVs can do a lot of things to a big chunk of DNA. And we have shown a while ago already that copy number variants were very well known in human developmental diseases, for example, in heart malformations or in the fetuses that are affected by a genomic disorder. We find a lot of CNVs as the cause, but CAKUT is just right up there next to the developmental delay and diaphragmatic hernia. and which was quite new at the time. So it's by right a human developmental disease, which is caused by in about 10% of cases a big deletion or duplication in the genome. And we've shown this in a large cohort of about 3,000 CAKUT patients from different ethnicities and also with different phenotypes, that CAKUT patients, they carry big chunks of DNA that are either deleted or duplicated compared to controls. So everybody has CNVs in their genome and they are responsible for variations. That's why we look all a little bit different. But in CAKUT patients, they are a lot bigger. So you can imagine that if they are bigger and more frequent, that they affect genes that are actually involved in, for example, nephrogenesis and lead to a CAKUT phenotype. And this is also what we saw because we know some very well-known pathogenic deletions like and duplications, like Julia said, for example, the HNF1 beta deletion at chromosome 17. Which is quite rare in children with renal hypodysplasia or kidney anomalies. So this drives this phenotype of a high proportion of deletions in renal hypodysplasia. But it also occurs in PUV that there are duplications that might drive the phenotype, which we know are genomic disorders. For example, Down syndrome is a very extreme duplication. Where you have a third chromosome 21, which is a duplication. And we saw this based on all these different CAKUT phenotypes in different proportions. But for kidney anomalies, this deletion kind of phenotype was quite well identified compared to controls, which is much lower. And again, what's also really nice is that if we have identified this known genomic disorder, for example, the 17q12, which is the HNF1 beta deletion, there are 6 big genomic disorders that explain about 2/3 of them, of the patients we found. So if we look individually at these big 6, as we call them, we see things that are quite interesting. For example, their locus specificity for HNF1 beta deletion in kidney anomalies, it's quite big, right? So you basically have a deletion there, whereas for PUV, those loci are more duplicated. So that's a different mechanism, different biological mechanism how this duplication or deletion comes to the phenotype. But there's also a lot of pleiotropy, which means that these deletions and duplications are found throughout the spectrum of CAKUT, which also explain maybe the variable expressivity of disease, as I already mentioned before. So we can learn a lot about understanding this disease based on the CNV data and also the complexity of it. Then we looked into more phenotype specific, and we found basically the same results for VUR, where CNVs are also important, and for obstructive uropathy. And these common suspects, they are also found there. So if you have a patient and you want to perform a relatively low-cost test for a genetic condition, then a microarray gives you, I think, a quite high yield, and it's relatively easy to perform. And this is also an explanation why some patients may have a more extensive phenotype, for example, with other extrarenal features in the brain or in the heart, whereas others only have isolated CAKUT. And it's all depending on how big the CNV is and which genes are interrupted or affected by the CNV, right? So the most easy model, and also the easiest model to validate, is a single gene CNV model, where if gene C is deleted or duplicated in the CNV, it leads to a phenotype, for example, in the kidney or the brain or the heart. But you can imagine that if the CNV is a little bit bigger and also encompass, for example, gene B and gene D, then you get a brain phenotype, whereas if only gene B and gene C are affected, you get a kidney phenotype. And based on this location and which genes are interrupted or affected, the extensivity of the phenotype is explained. But you can also imagine that it's much harder to validate such mechanisms in, for example, mouse, which have a different genome and also different arrangement of genes, or a fish. So proving causality in these more complex models for CNV is much harder than for a single CNV model. So the easiest way is just to go for the monogenic cause, and we have done this for many of these more common genomic disorders. So for 17q12, it's H and F1 beta, for 22q11.2, so the D. George locus, we have shown that it's Crickle, and for the 16p11.2 microdeletion syndrome, which is very common associated with autism, we have shown in the mouse model that the TBX6 can drive this gene, this kidney phenotype, whereas others have shown that there's another gene that's quite close to TBX6, which is MAS, can lead to a genital urinary disorder. So it might have two drivers for the CAKUT phenotype, and it comes more into the oligogenic type of disease. And what's also nice, this is a quite recent publication from a group from Serbia who showed that CNVs are not only encompassing genes, but can also harbor microRNAs, which are these modulators or regulators of gene translation. And if these genes are, or if these mRNAs are affected or deleted, for example, it can change gene expression, for example, in this case from NOTCH3 in cells, and NOTCH3 is a very common gene known in CAKUT. And if you have a CNV that interrupts this microRNA, the expression of NOTCH3 is a lot lower, and which might then affect your CAKUT phenotype. So these CNVs can also learn us about the mechanisms through not the epigenetic mechanism, so to say, and not only the genetic mechanisms of CAKUT. So this really requires more extensive work to prove causality, but it opens up new avenues to identify the etiology of CAKUT. And then now we are doing quite more whole genome sequencing. For example, this is data from Melanie Chen, who's in London, who performed the whole genome sequencing study in children with posterior urethral valves. And with this whole genome study, you can do a GWAS, but you can also look into CNVs, and you can look into monogenic causes. And this whole genome sequencing also gives in-depth information about not only deletions and duplications, but also inversions, right? So if the DNA is flipped, for example, and that can disrupt the gene or cause the phenotype as well. And she found that inversions are actually quite common in PUV. So if you do genetic testing in CAKUT, don't only do a gene panel, a whole exome sequencing gene panel, so to say, but do not forget the CNVs, and they become more easily to do also in sequencing data, especially in genome sequencing. And this may help to get this genetic diagnosis and to end diagnostic odysseys of patients, but they also are a springboard towards better treatment of patients, and also to identify extrarenal features that have not come to light before. For example, the father with the three phalangeal thumb, which he knew, but no medical specialist would care about. So this picture Julia also showed about the common variants in CAKUT, and which are, I think, more susceptible to CAKUT phenotypes that are not rare at all. For example, vasculourethral reflux, duplex kidney, common variants that have a low effect size, but together, so polygenic, so to say, cause the phenotype, might better fit the disease model than these rare monogenic variants. And we need the genome-wide association studies for this. to find them. But the problem with GWAS right now is that we don't have big enough cohorts. You need, so to say, more than 10,000 patients and controls to identify these associations, because you have to have lots of patients who have the power to prove genome-wide significance to find them. And the other problem, of course, we have with GWAS is that we don't really know the clinical significance. So if we find a common variant that is associated to the disease, yeah, we don't really know, are they truly causal? They're most of the time they're not in the exome of a gene, but in the intron or even intergenic. So yeah, what is then the clinical significance, right? If we find a statistical association, will it actually help the patients? But these finding these common variants, it's a starting point of understanding the disease, and also yeah, better studies to improve the outcome. So there are not many GWAS in CAKUT because our yeah, the groups are very heterogeneous and not big enough, and there's only a handful right now. And I think one of the things we need to do is we need to collaborate in the world to make sure that we at least get the numbers and more homogeneous group, and that we can perform very relevant GWAS. And you can also do GWAS, for example, to calculate polygenic risk scores, so genetic scores that gives you an indication about the risk of developing a disease or developing complications of the disease. For example, many CAKUT patients, they have a mild CKD phenotype, and only the tip of the iceberg, they will go into kidney failure. They are a very relevant group in pediatric nephrology, of course, because they're severely affected, but they don't represent the entire CAKUT spectrum. And I think there are also a lot of adult patients with CAKUT who don't really know that they have CAKUT, for example, because they never have an ultrasound in their lives, who actually live a quite normal life. And it would be great right now if we, because we do all these antenatal ultrasonographies and we diagnose the patients early, that we actually can predict the outcome, for example, based on genetic risk profiles. So this is the future maybe, but we should work on this to get there and actually help our patients, in my opinion. So one of the ways we in the Netherlands try to do this is by founding the Ardeco Consortium, which is a nationwide effort to biobank all the patients we know with CAKUT and get their DNA, get environmental hazard exposure by questionnaires of mothers about their pregnancies, and also link these to their clinical data, so we can actually start trying to establish genotype-phenotype associations. So how we do this in the Netherlands, we are helped by the Dutch Kidney Foundation and by EURECA and Eurogen, but we collaborate in the 6 big pediatric nephrology university hospitals to collect all these patients, and we have now gathered about 3,500 CAKUT patients with all phenotypes, rare and common. We have their DNA and of the DNA of their parents, and we have their clinical data and environmental data, and we are now currently performing studies to find them. So, for example, by doing a GWAS in posterior urethral valves with whole genome sequencing data, we collaborate with London to find these and identify SNPs, common variants that are associated with CAKUT. You get these Manhattan plots, which look a little bit like the skyline of my favorite city in the world. But you see, we need bigger numbers to get above this red line, which is the line for genome-wide significance. So we need to expand our cohort, and I'm happy that, for example, we now work together with Alina to actually get there. And then we are also doing a CAKUT family study by using GWAS data from whole genome sequencing, which we do association testing in the parent-patient trios. So we look which allele is given to the affected patient by the parent, and if we see a pattern that prefers one allele above the other, then we know that this might be a very relevant allele in the etiology of CAKUT. And we try to do this across the entire spectrum of diseases which I talked about. So this is work in progress from my PhD student Lisanne Vendrich and Michel Tank. And we found actually quite some suggestive loci in this whole genome data. And now we are expanding our cohort by adding new patients and see if we can get them to a genome-wide significance. So next time that you invite me to give a talk, I can hopefully provide you with the data to show what the etiology of this trio-based study is. So with that, I come to my conclusion. So CAKUT is characterized by a heterogeneous clinical presentation and molecular basis which provide a rationale for a complex etiology of this disease. It is tremendously challenging to find this complex background, but I think it's very relevant to do because we may can better help and predict the future of these patients. Structural variants and common variants, they are important in CAKUT, but we need large-scale collaborations to find them. And we are very fortunate within the ESPN, but hopefully also in the World Kidney Academy to set up such collaborations, such as the EURECA or ARDECO studies, to see if we can actually come to important results. And with that, I want to thank you for your attention.
Thank you very much, Rick. I really highly enjoyed both of you, the talks of both of you, Julia and Ulrich. It's really a very terrific talk, especially in the beginning. Give the basic about the genetics. I always talking about genetic. As you know, for the last last 10 years, the high advance in genetic testing, it make it very easy for us as a clinician to reach out and point the diseases and renal diseases, whether pre or post kidney transplantation. And always I'm talking about this for the two things that became very advanced: the genetic testing and point of care ultrasounds. That probably we need to teach our medical students and give him a lot of teaching about these advanced technology that help the clinician about it. Before we start reading the question, I would like also to thank the European Society of Nephrology of joining us in World Kidney Academy to give this wonderful talk. So we'll go over some of the question. Handsome, she said that in case of CKD, we don't group CAKUT and genetic causes separate. Are genetic kidney disease such as autosomal dominant polycystic kidney or autosomal recessive, etc., considered as a CAKUT, and what is the distinction? So Rick is.
Yeah, I can answer that. So yeah, so what you actually put into the spectrum of CAKUT is quite up for discussion still, right? But the more or less scientific community puts in the kidney anomaly phenotypes, urethral phenotypes, and some include the bladder or lower urinary tract phenotypes, whereas others are opposed to this. I think ADPKD is a very distinct phenotype and also has, of course, a better described genetic etiology of PKD1 and PKD2 mutations. But sometimes the distinction with multicystic dysplastic kidney is quite hard, right? So in PKD, you see the cyst appear and the kidneys enlarge, whereas for MCDK, you see multicystic dysplastic kidneys, which look quite different. But this is a common thing that gets switched in the discussion. So I won't consider PKD as a CAKUT phenotype, but as a distinct genetic disease, and it's also more a disease, although it occurs in children, which our adult nephrology colleagues see more often than we do, at least the effects from it.
Totally agree with you.
And I think maybe also from an embryology point of view, you can say that the CAKUT spectrum always arises from defects that arise from the Wolffian duct and ureteric budding. And this is also why, for example, the bladder is bared out, but the posterior urethral valves, which occur at the point where the Wolffian duct insists into the anorectum or the cloacal tissue, are included, but bladder deficiencies like bladder exstrophy are not included into CAKUT. So I think this is kind of, if you see it from an embryology point of view, why ADPKD and RAPKD cannot be considered CAKUT.
Okay, thank you very much. Ben, she's asking, what the meant by monogenic? Doesn't mean there is a pyogenic? Give me pyogenic example and epigenic as well. I think probably Julia, you give a little bit and explanation about this. Would you like to answer, Julia, about this? Julia, are you with us?
Yep. Sorry.
Yeah, go.
I think I answered this in one of the first pictures. So monogenic, you have one gene where you have a variant. Polygenic means you have several genes affected where are variants inside and which interact and lead to a phenotype. And epigenetic means that you have a change in the methylation, but at least to my knowledge, there are only a handful of data in the literature available which examine epigenetic features or changes leading to CAKUT phenotype. I think also this needs larger studies on this effect, but I for sure think that epigenetic factors are a cause or maybe an important cause for CAKUT.
Okay, thank you very much. I'm seeing a question from Raja Abdurrahman, probably is not for CAKUT, but for genetic in general. So any genetic association between psoriasis and nephrotic syndrome, if both presented in the family. So probably an example of renal and extrarenal, but it's not CAKUT. So would you like Rick to answer this?
Well, I mean, they're both considered immunological conditions, right? So I'm not aware of a genetic cause that affects both of them. But podocytopathies, there are also, just as CAKUT, many different genes involved. And we also see a lot of, for example, hay fever or allergies, eczema in children with nephrotic syndrome, which has not been explained too much. But yeah, I don't know a clear genetic cause that causes psoriasis and nephrotic syndrome out of my head, at least.
Thank you very much.
But maybe it would be helpful just to perform probably exome sequencing to look, or a panel diagnostic if you find a disease-causing variant in a nephrotic gene. And yeah,
that's which one.
Especially if the patient is steroid resistant, for example, right?
Yeah. You know, well, nephrotic syndrome a little bit, it's confusing a little bit. So we don't know exactly the pathophysiology. So it's not only the immunological reactions or autoimmune, reactions, but as Julia, she mentioned, there is some genetic disease, well-known genetic mutation that can cause a specific types of nephrotic syndrome. Another question. Yeah, that's a good question from my friend Rajiv Sinha. He said, do you have to do the number, the CNV variant, the number of variants, even if you have done whole exome sequences? Maybe I will add, what about also for whole genome sequencing? So either of you who would like to answer, Rick or Julia?
So it depends. I can start, and maybe then Julia can add. So it depends a little bit about your bioinformatic skills or your help in this, right? So if you do a whole exome sequencing right now, you can with some tricks you can actually look into CNVs quite well, but it requires bioinformatic skills to do it. For whole genome, sequencing, it's a little bit easier. So hopefully with the large rollout of whole genome sequencing, you don't have to do a separate chromosomal microarray to find the CNVs, and you can do it with one modality. On the other hand, a DNA microarray is not that expensive anymore. In research setting, we do them, for example, for 30 euros per patient. So it's quite cheap compared to trio exome sequencing or whole genome sequencing.
Excellent, excellent. I thought the whole genome sequencing is probably you don't need to do it. Is that right? Or I'm confused.
If you want to look into CNVs, it's you can do it with whole genome sequencing, and also you can do a GWAS with whole genome sequencing, and you don't need imputation, which you need to do with DNA microarray. So it's actually, yeah, I think it is the future. Yeah.
So I think due to the time, we are already a little bit over, but it was so interesting, all your talks and the questions. But I think we need to finish up now. Niveen already has left us. So Julia already shared in the chat her email address, and I did share the email addresses of Rick and myself as well for the questions that now have been left unanswered. And also there was one question asking whether collaboration within our ongoing studies is possible, and Julia already, I think, answered that for the EURECA registry, and Rick already made his point for the ARCH-DECO study, and for patients with posterior urethral valves. I have the Care for Lupus study.
Alina, if you allow me, just last the question. So as I'm handling the questions. So the questions to Julia and Rick. What about doing in utero genetic testings especially for families that they have known cases, they have kids with genetic diseases, renal genetic diseases. So what about doing in utero, especially very early time during a pregnancy?
I think this depends on the phenotype of the child. I think it's not possible to answer the question just for all patients. I think it's possible to do genetic testing, at least in Germany, during the 11th and 12th week of gestation. There you can perform prenatal testing, and then you also are allowed to stop or to terminate the pregnancy if the genetic testing showed a child which will be severely affected.
Excellent. Thank you very much. And probably later on will actually go hands to hands with the pre-implantation diagnosis and to avoid all these renal genetic diseases. The mic's with you. So thank you very much. Thanks. The mic's with you, Alina.
No, that was indeed a really important question. And I think for all the questions left unanswered, you are free to send emails to Rick and Julia and ask your questions. And I would like to thank the organizers for this great workshop and this really nice cooperation with our ESPN CAKUT working group. And thank you all for attending.